Greetings, my name is Ian, and today I'm going to talk
Talk to the microphone.
I'm going to talk about in vitro activity of essential oils against the Saprolegnia parasitica. And at first, we have to know what's in vitro. In vitro means that it's a kind of performing procedures that in controlled environments, especially outside of the living organism. And there's a word about outside words. It's so-called in vivo. It means that you're doing some research and inside of the body. And we also have to know what's essential oils. It's a kind of liquid that containing volatile chemical compound from plant. Most of the essential oil just smells good, just like flowers, butterflies, gardens, etc. Just like the dolphin. And what's Saprolegnia parasitica? It is a kind of water mold that severely impacts fish health in agriculture. We can just look at a picture. It is the fish. The surface of fish is surrounded by the slick-like mycelium. And I can tell you that this fish is so dead. Yeah. And if you still don't know what is Saprolegnia parasitica or oomycetes, you can just think about it. You are in group, and one of your members he just don't participate in anything, and you can just go and tell him that even the worms in my stomach are less parasitic than you. Just joking. And the purpose of the research, we had to know that water mold had severely impact fish health, and we also know it will sabotage its eggs. But the important thing we have to know, there is no drugs license for treating or control the organisms. So it means that once the fish infects the parasitic oomycete, then you have nothing to do. Just watch its life fading away slowly. So, to solve the problem, we have to find a new map to control such infection. The purpose of the research is to make the agricultural booming, to make the agricultural flourishing, and together to make the agriculture great again. Yeah. And so we use GCMS analysis to know every single component of the essential oils. So we can see the x-axis is the essential oils and the y-axis is its component. And I will explain what is GCMS analysis later. And from the data, we can see OV and TV are at the minimum inhibitory concentration. That means these two essential oils is the most effective. And OM is really impressive, but it's not as good as OV and TV. And CB, CLO, CLC, these three essential oils are at the rate of 2.5. And CLCP is not really useful. Yeah. And its rate of about 10. And this is the raw data. At first I saw that, I think, wow, gosh, it's really difficult. But when you just look into its information and try to clarify, try to know what's going on. And in fact, I know that, wow, I really hoisted by my own petard. And so we have to know that the most effective essential oil is T virus and O Bulgaria. Yeah. And the chemical composition of this LC is similar to CBCF. Remember that is O of R 2.5. Minimum inhibitory concentration. Yeah, and we just look at this data, we know that OVTV are most effective, but why? So we just look into it and we know that this thymol and carboxyl are the most important components that will make the TV and OV so powerful and so strong. But it also have something problems. These two components sometimes will make fish sick, make fish weak. And when we eat this fish with thymo and coverall, and we get some problems just like diarrhea, bovine, or some chromomic disease like cancer. So these three essential oils, I think it is not suitable for put it into the agriculture. And we have mentioned that CP, CF, and LC is also really good. Yeah, and the chemical composition is same, 2.5. Not same, very similar. So I told that C. Bergermia is the most unique among other species essential oils. We can just look at its component, C. Bergermia, and his luminance is 33.2. You just ignoring OM, OV, and TV. We can just look at limonium. It's relatively low when comparing CL and CP. We can just see these two essential oils are not useful because its inhibitory constant, minimum inhibitory concentration load is literally high. And it's the non-relevant. And it's the non-acetate. Both of these components is really high when comparing with the other essential oils. And also I have to mention that its monoterpene hydrocarbonate and its oxygenic monoterpenate is really similar. Yeah, it is so special. This researchers find so many essential oils, just this one have this characterized. And we know that the Saprolegnia oomycetes are characterized by the nature resistant to anti-mycotic drugs. It means that it is hard to kill due to the peculiar silver composition method of the silicic and glycan. Just think about the disease that happens in human, the oomycetes. It just infect our feet. And his mixing will just root into your feet, and make you feel itch, make you feel pain, and make your foot feel odor, feel bad. This is so-called authentic feet. You can also call it Hong Kong feet, just like that. And his bacterial and method, all of the essential oils I mentioned about were purchased from the Italian shop. It's called Ferrara shop. I have no idea. And volatile constitutes of each word analysis by GC-MS analysis. GC for gas chromatography and MS for mass spectrometry. It is kind of an analytical map to identify different substances within a test sample. So from the beginning table, you can just see every single component of the essential oil. And in vitro exit, we know that figure the minimum inhibitory concentration, right? And actually, this experiment is due to test the rainbow trout. It kind of disease that happen in fish. The researchers just put the essential oils to this rainbow trout and to look into its utility. And then this rainbow trout, he just mentioned, it's just maintained in milk extract broth. It is kind of water that can keep microorganisms live, keep it reproduce. And we also know that the lowest concentration at which function growth is not noticeable. Because the data indicate that minimum in inhibitory concentration and minimum fungicide concentration are corresponding. The lower the MIC rate and the higher the anti-fungicide, the higher the fungicide. It means that the lower the rate, it have the good effective to fight the virus. In conclusion, finally, we know that LC and CF and CP, these three essential oils, is minimum inhibitory concentration rate is 2.5. It is the most promising essential oil to fight the virus. Yeah. And the last but not least, I have to say that I think this paper is not really comprehensive, because it just mentioned that which one essential oil is the most effective and which one isn't. But it didn't tell you why and how. So at my next presentation, I will go find more information and just try to give you a reasonable explanation. So thanks. It's my presentation. Viva la vida. Hope you have a good day.
Okay, 11 minutes 40 seconds. Rina. Echo. Echo. Ethan. Ethan. Rich. Sarah. Okay, Rina.
Pardon?
Your slide is CL or something else.
Which one?
Your previous. Have this. In X-axis, you mentioned about CB, CL, etc. And can you explain more about the differences between them?
You can just look at this data, and the CP is limonene is the relatively low when comparing CL and CP, and its linoleucine is relatively high when among with these essential oils, and its linoleic acetate is really high. You can see there is no other essential oils is surpassed than CB.
We move on. Next, Ethan.
My question is the infection. When does this infection happen most frequently?
You mean the Saprolegnia parasitica?
Yes.
This virus, it usually happens in winter because it like the relatively low temperature. Yeah, the low temperature that makes it feel like reproduce.
Rich.
So I want to ask about what is rainbow trout.
Rainbow trout is kind of the disease that happens in fish. It's kind of virus that happens in fish. They will make fish sick. And sooner or later,
Rainbow trout?
yeah, kind of
Is
virus.
It's a virus?
Virus, yeah,
Sure?
definitely. Yeah.
Sarah?
Yeah. 红彩病毒吗 ？Rainbow trout. Virus
Rainbow
never.
trout is a kind of fish.
Kind of virus never make fish sick.
Really?
Yeah, I think so.
Let's move on. Sarah.
Can you go to the discussion? I want to. The glycan means what? Glycan means what?
Lichen. I know his Chinese word, I'm not really good in biology. So it kind of made of the glucose. Maybe.
Volunteer question, Eddie. Volunteer question? One? Any more? Two. Okay, Sam? Yeah.
So what's the adaptation of these results?
Application?
Yeah. What the result can apply on what the result can apply on apply? Yeah. What the result can tell us? Yes.
Result.
Yes.
This. And we know that even know that OV, TV, and OM, it is the most effective essential oils that can put in water to fight against the virus, but it also has some toxic. So we just pick CB, CF, LC. It is also good to as a fungicide.
Yeah.
What is monoterpene?
Monoterpene.
Yeah.
You want a simple explanation or a
Simple.
just like benzophenone? It's a kind of lipid, but it can dissolve in water. It is organic compound, and just like benzophenone. A mono means one or single, and tetra means benzophenone.
Okay. One second. You have to stay. Okay, so I think the strength of his talk was he talked really fluently and in a competent manner. But I think there are some things that can be improved in his talk. At the later part, let's start with introduction. So at the later part, you started to talk about Hong Kong food, something like that. I would say this is something that can be put in introduction because most of us, this is a disease, and if you can draw our attention by telling us we have similar disease in humankind, then that will give us a better understanding of what kind of disease you are talking about. And then you can say that in humankind there are some gel that you can put on your food to cure this disease. But in fish, this is really, really difficult. So far, there's no cure for it. That's the reason why the authors want to try these essential oils. So that's something that can be improved in introduction. Then after introduction, this is a nice purpose slide. Then you go directly to your results. Is it the structure of the paper? they have material method at the end of the paper? Papers? Because results. Okay, so usually I would say the results and discussion, although in some papers this might be at the end, because many people think this is technical details. But I think for this class, it's necessary that you have to present it right after introduction, especially when you talk about GC or whatever is not that important. But the most important thing is minimum
Inventory concentration.
MIC. Yeah, you have to explain it clearly because you present result first, and we always think a higher MIC might be good or whatever. Yeah, you probably need to tell us what kind of MIC you and the researcher want to see. Apparently, you want a lower MIC, right? Yeah, you said, but it's not right at the first place where you show it. Okay, so you have to show it, say it clearly that this. Ideally, it's better that you present it first, and then you discuss what kind of results you are expecting. Then and try to talk about material method. And the material method is a little bit wordy, so try to condense the text. Then to the results. Okay, so this is a self-made figure, which is nice. But again, like most of the self-made figures I have seen so far, the main weakness is always the font size. So please remember you can adjust the font size in Excel. And okay, so the unit is here. Usually we tend to put it next to the label. So I was looking for the axis label. Okay. And then here you have different species of essential oils. Because at the end you have some criticism of this study, which I very welcome. But I wonder if, when talking about the study itself, could you please provide us more information about these effective essential oils? For example, here you just talk about its Latin name. Do they have common name, or do we see these plants in Taiwan or whatever? That's my question, because this, I personally have no feeling to these essential oil or these plants. What are they? So maybe you can tell us more about these plants. Okay. Then at the end, you gave us some criticism of this study, which I very much agree. So next time, if you want to add something that tells more about the mechanism, I look forward to it. So let's thank Ian. This is the end of all talks 1. And starting from next week, we have talk 2. And let me remind all of you again, the spirit of talk 2 is I need to see your improvement. Okay. No improvement means your score will be lower than your talk 1. And talk 2 is 45%, almost half of your semester score. So next week we have Sky, Lina, Alan, Sam, and Young. And for those who wants to add subtitles, you can only do that before your talk 1. And after your talk 1, you are no longer allowed to add subtitles. Okay, class dismissed.
所以我变成大福利站。
是啊。
还是给你准备海绵便当。
讲这样快了，还超过时间了，现在厕所关了。
你要上过日常作业，是那一份你是这样可以但是你要能够在笔传上作业，就是那份给你去写，没有抄。但是事实上剧是不行，可是要一个那个就是有效的传作业的，因为它是
可是我觉得我应该应
证
就是看
你要有
我遮完那个干嘛？可是我
不是这个，如果你有潜水证照的话，就可以做。是我知道。
那到时候如果我们有钱的，就是拿潜水证
理论上是的，不过因为我们以前去龟山岛我都是用潜水证，我没有上过你的课，所以我知道潜水证是可以的。
但是到时候海巡说我改会说什么的？
理论上应该是不会挡吧，但是你当然可以在事前确认。你有跟他说你有潜水证照吗？
今天在跟那个海巡的讲一下。
对，你跟他说你是 party 吗？还是什么
？party 的。
对，应该可以，因为我也是 party。潜水证是我知道是一定可以。
好，比较。
喂。